Assalamu alaikum everyone. In this video lecture, we will be discussing the paper 1 topical past papers of the chapter Enzymes from AS level biology 9700. Let's start. MCQ number 35. Two experiments were carried out using an enzyme from humans. The first experiment X was carried out at a constant temperature of 37 degrees Celsius. So you guys know that 37 degrees Celsius is the optimum temperature of most of the enzymes in the human body. During the second experiment, the temperature was increased from 37 degrees Celsius to 80 degree Celsius. All the factors were kept the same. Right. So in the second experiment, they have increased the temperature from 37 degrees Celsius to 80 degrees Celsius during the experiment. And in the first experiment, which was the experiment X, in that experiment, they kept the temperature constant to 37 degrees Celsius. Which graph shows the results? Now guys, look at these graphs. There is product concentration on the y-axis and time on the x-axis. So if we look at the first experiment or experiment X, in this experiment, product concentration will be high. The final product will be high because this reaction had a constant temperature of 37 degrees Celsius and the enzymes were working at their best. So the reaction will be completed and all the substrates will be converted into product. So the final product concentration will be the highest, right? If we talk about the second experiment, during the second experiment, temperature was increased to 80 degrees Celsius, which causes the enzymes to denature. So the reaction will not complete fully because the enzymes will denature and the product concentration or the products formed in the second reaction will be less as compared to the amount of products formed during the first experiment. So the answer will be A, because due to denaturation of the enzyme, the reaction will stop quickly as compared to the first experiment. In first experiment, the reaction is completed at around 3 minutes, whereas in the second experiment, the reaction will be completed around 2 minutes because the enzymes are being denatured and less products will be formed because the enzymes are denatured quickly. The B cannot be the answer because in B, the product concentration is falling. The product concentration will not fall, right? Because when the enzymes are denatured, the product concentration becomes constant because products are not being made. And this is not the case that product concentration will fall because products are not being used, right? C cannot be the answer because in C, the product concentration of the first and the second experiment, the final product concentration in both experiments are equal, and this is not possible. The product concentration in the second experiment should be less because enzymes are denatured. And D cannot be the answer because again over here the product concentration is falling and this is not the case. Product concentration always rises in an enzyme experiment, right? And if enzymes are denatured from the start, the products will not be made, the product concentration will be 0. And if enzymes are denatured during the experiment, so some product will be formed and the product concentration will always rise. That's why the answer is A. Let's move on to MCQ number 34. Following a heart attack, the enzyme lactate dehydrogenase leaks into the blood plasma from the damaged heart muscles. Which steps are required to obtain the estimate of lactate dehydrogenase activity? So guys, we have to obtain an estimate of the lactate dehydrogenase activity, or we can say we have to find the rate of lactate dehydrogenase in a sample of the blood plasma, right? So which steps are basically required to obtain this estimate of lactate dehydrogenase activity? So guys, if we want to estimate the lactate dehydrogenase activity in the blood plasma, we need to incubate the lactate dehydrogenase with the substrate. So this should be a tick. And sterilize blood plasma by heating. We don't need to heat the blood plasma because if we heat the blood plasma, lactate dehydrogenase will denature and we will not be able to obtain the estimate of the lactate dehydrogenase activity. Incubate with lactate dehydrogenase inhibitor. We should not do this because if we add inhibitor, the activity of the lactate dehydrogenase will decrease and we will not obtain a proper estimate of the lactate dehydrogenase activity. So the answer is C. So guys, by this method we can measure the amount of lactate dehydrogenase leaking into the blood, and this can indicate the extent of heart attack or heart muscle damage, right? So for example, if a large extent of heart muscle is damaged, so a lot of lactate dehydrogenase will leak into the blood, and there will be high activity of lactate dehydrogenase in the blood plasma. So if you want to measure the activity of lactate dehydrogenase in the blood plasma, don't use inhibitor, don't heat the plasma, just incubate with the substrate, and you will know the lactate dehydrogenase activity in the plasma or the concentration of the lactate dehydrogenase in the blood plasma. Let's move on to MCQ number 33. The graph shows the effect of increasing the concentration of substrate on the rate of an enzyme catalyzed reaction. So guys, this is the graph of concentration of substrate on x-axis and rate of reaction on y-axis, right? And this is a normal graph without the inhibitor, and graph 3 is with the competitive inhibitor, fixed concentration of competitive inhibitor, and graph 4 is a graph with fixed concentration of non-competitive inhibitor. What is limiting the rate of an enzyme catalyzed reaction at 1, 2, 3, and 4 on the graph? So guys, 1 and 2 are the points on the graph without the inhibitor, right? So at 1 we can see that as we are increasing the substrate concentration, so at 1 the rate of reaction is increasing. It means substrate concentration is limiting the rate. Whenever you increase the concentration of a limiting factor, the rate will increase. Or we can say that if we increase any factor and the rate of reaction increases, it means that factor is the limiting factor. So at 1, substrate concentration is the limiting factor because as we are increasing the substrate concentration, rate is increasing. Substrate concentration is low at 1, that's why increasing the substrate concentration increases the rate. If we talk about 2, so guys, at 2, when we are increasing the substrate concentration, there is no change in the rate of reaction. It means that substrate is in excess and enzyme concentration is limiting. So guys, at 2, enzyme concentration is limiting the rate. At point 3, the rate of reaction is low as compared to the original graph. So at 3, for example, the rate of reaction should be at this point, but it is lower, and that is because of the competitive inhibitor, because at 3 the competitive inhibitor is added. And if we talk about 4, so guys, at 4 the rate should be over here, but the rate is lower, that is because of the non-competitive inhibitor. So at 4, non-competitive inhibitor is limiting the rate, at 3 competitive inhibitor is limiting the rate, and at 1 substrate concentration is limiting the rate, and at 2 enzyme concentration is limiting the rate. So answer is C. Let's move on to MCQ number 32. Which row about competitive inhibitors of enzymes is correct? Binds to a site other than the active site. Guys, this is not true for competitive inhibitors because competitive inhibitors, they bind to the active site of the enzyme, and it's the non-competitive inhibitor which binds to the site other than the active site and lower down the activation energy needed for the reactions to occur. So guys, competitive inhibitors do not lower down the activation energy. The answer is D. Activation energy is reduced by the enzyme and not the competitive inhibitor of the enzyme. Let's move on to MCQ number 31. Influenza virus has an enzyme called neuraminidase, which breaks down glycoproteins in the membrane of the cell that the virus will infect. The glycoprotein binds to the active site of the neuraminidase by induced fit. Which statements about the induced fit hypothesis of an enzyme action are correct? The active site must have a complementary shape to the substrate for them to bind together. Guys, this is wrong because induced fit hypothesis states that the shape of the active site is not exactly complementary to the shape of the substrate, and when the substrate fits or binds, it induces a slight change in the shape of the active site for perfect fit, right? So the shape of the active site is not exactly complementary to the substrate, as far as induced fit model is concerned. So statement 1 is wrong. Let's look at the statement 2. This enzyme is less likely to be affected by non-competitive inhibitors than an enzyme working by the lock and key mechanism. Right. So guys, this statement is wrong again because lock and key hypothesis states that the enzymes are very rigid and they don't change shape. So if the enzyme is working by lock and key mechanism and the non-competitive inhibitor binds, right, the shape of the enzyme will not change because the lock and key hypothesis or lock and key mechanism says that the enzyme is rigid. So the non-competitive inhibitor will less likely to affect the lock and key mechanism enzyme. Are you getting my point? I'm repeating this again, that lock and key mechanism enzyme or the enzyme following the lock and key hypothesis is rigid, and if the non-competitive inhibitor binds, that enzyme will not change shape. So the active site is less likely to be altered. The shape of the active site will less likely to be altered. Right? So the enzyme following the lock and key mechanism is less likely to be affected, and the induced fit enzyme will more likely to be affected because the induced fit hypothesis says that the enzymes are flexible, and when the non-competitive inhibitor will bind, the shape of the active site will change and the shape of the enzyme will change. So this statement is false, right? Let's move on to statement 3. The substrate is converted to product by specific R groups in the active site, just like the lock and key mechanism. Guys, this is true. Whenever the substrate fits into the active site, there are specific R groups in the active site which make specific interactions with the substrate that allows it to convert to the product. And this is true for all the mechanisms, whether the lock and key mechanism or whether induced fit mechanism. So answer is D. Let's move on to MCQ number 30. A fixed volume of the enzyme catalase was added to a fixed volume of hydrogen peroxide. So guys, enzyme is the catalase and the substrate is hydrogen peroxide. The diagram shows how the amount of product changed over the course of the reaction. So guys, we have time on x-axis and amount of product on y-axis, and we can see that as the time proceeds, the amount of product increases and it becomes constant. What explains the shape of this graph? The active sites become saturated? No, this is not the case, because if the active sites become saturated, the rate of reaction becomes constant and not the amount of product, right? If the reaction is occurring at a constant speed or a constant rate, the products are made at a constant speed, right? The amount of product will not become constant, the amount of product will increase constantly. You need to understand this, because if the rate of reaction becomes constant, products are still being made and the product concentration will increase, right? Always remember this point. Whenever the active sites become saturated with the enzyme, it's the rate that becomes constant and not the amount of product. Amount of product continues to increase because the rate of reaction is constant and not zero. Whenever the rate of reaction is zero, right, then the products are not being made and the product concentration will become constant. The enzyme was denatured. Guys, we don't have any clue in the question. We don't have any information in the question which says that the enzyme is denatured, right? They did not tell us about the temperature or pH or anything or any change during the reaction. The hydrogen peroxide inhibited the reaction. Guys, hydrogen peroxide cannot inhibit the reaction because hydrogen peroxide is the substrate of the catalase. It cannot inhibit the catalase. The substrate molecules were being used up or were used up, yes. Because the substrate was used up, the reaction stopped and the product concentration or the amount of product became constant. The answer is D. MCQ number 29. Which statement about enzyme inhibitors are correct? Competitive inhibitors may be similar shapes to the substrate. This is true. Competitive inhibitors have identical shape to the substrate. Competitive inhibitors bind to the active site. This is again true. Non-competitive inhibitors alter the shape of the enzyme. They do. Non-competitive inhibitors bind to the substrate. No guys, they don't bind to the substrate. Non-competitive inhibitors bind to the allosteric sites, which are sites other than the active site. So the answer is A, 1, 2 and 3. The graph shows the energy levels involved in an enzyme catalyzed reaction. Substrate molecules X and Y combine to give product Z. So guys, X and Y combine to give product Z. Which arrow shows the reduction in the activation energy due to enzyme? So guys, C is the activation energy of uncatalyzed reaction and A is the activation energy of catalyzed. So guys, we need to find the reduction in the activation energy due to enzyme. So initially the activation energy was C, and due to enzyme the activation energy becomes A. So C minus A over here is B, right? We can see that this is C, and if we subtract A from C, we get B. So the B is the answer. Let's move on to MCQ number 27. Lipase is a digestive enzyme produced by the pancreas that catalyzes the hydrolysis of dietary lipids. The table shows how the pH of a liquid food containing a high proportion of lipids decreases over time. So guys, you need to know that lipase is an enzyme which breaks down lipids into fatty acids and glycerol, and fatty acids, which are the products of this reaction, are acidic in nature, and they will cause the pH of the liquid food to drop. So guys, whenever lipids will be broken down by lipases, fatty acids will be made, and fatty acids will decrease the pH of the solution, right? So over here we can see that pH is decreasing with time, and the pH then becomes constant. And why is that so? Why does the pH becomes constant? Maybe because the decrease in pH denatures the lipase enzyme and the reaction stops. So guys, if the reaction keeps on going, for example, if the reaction keeps on going, the pH will continue to fall because fatty acids are being produced, right? So higher the concentration of fatty acids that are being produced, lower the pH, right? So the reaction of lipase will always show fall in pH, and when the pH falls too much, the lipase denatures and the reaction stops. The reaction can also stop maybe because the substrate concentration can be limiting, or we can say because the substrates are used up. So reaction can stop either because of the use of the substrate, or either because of the denaturation of the lipase enzyme, right? So which statements are possible explanations of the results of the experiment between 50 and 60 minutes? So guys, the reaction between 50 and 60 minutes shows the constant phase of the graph. So over here we need to select the statements which are best or possible explanations. Enzyme concentration becomes the limiting factor. No, enzyme concentration is not the limiting factor over here, because if the enzyme concentration was the limiting factor, the reaction would still be going on and the pH would continue to fall. The pH, when it becomes constant, it means that the reaction has stopped. Substrate concentration becomes a limiting factor. This is true. That's because the substrates are being used up. All the enzyme active sites are saturated. No. If all the enzyme active sites were saturated, the reaction would still be going on. Denaturation of the enzyme by the products. This is true, because the product over here is the fatty acids, right? Products are the fatty acids, and these fatty acids, they are decreasing the pH, causing the denaturation of the enzyme. Products are acting as competitive inhibitors. This is possible, guys, that products which are formed may act as the competitive inhibitors of the enzyme, and this can slow down the reaction or even may stop the reaction. The answer is 2, 4, and 5. The answer is D. Let's move on to MCQ number 26. Which words from the table correctly complete the paragraph about enzymes? When the pH of an environment is decreased below an enzyme's optimum pH, dash bonds between adjacent dash groups holding the dash structure are disrupted. So guys, whenever a pH is decreased below the enzyme's optimum pH, always remember that hydrogen and ionic bonds are broken, right? Hydrogen and ionic bonds which hold up the tertiary structure of a protein are broken down by changes in pH, right? So the hydrogen and ionic bonds between adjacent R groups holding the tertiary structure of the enzyme are disrupted. So the answer is A. Let's move on to MCQ number 25. Ethylene glycol is a chemical used to prevent water from freezing. If ethylene glycol is swallowed accidentally, it is metabolized by an enzyme found in the liver cells to produce a toxic product. The enzyme normally catalyzes the oxidation of ethanol to a harmless product. So guys, there's an enzyme which normally catalyzes the oxidation of ethanol to a harmless product, and the same enzyme can metabolize the ethylene glycol to a toxic product. People who have swallowed ethylene glycol are treated with large doses of ethanol. This prevents formation of a toxic product and allows the body to excrete the ethylene glycol. So guys, if somebody has accidentally ingested or swallowed ethylene glycol, right, they can be given ethanol because the enzymes which metabolize ethylene glycol and ethanol, they both are the same enzymes. So what will happen is that if we give large amounts of ethanol, the ethanol is more likely to bind to the enzyme and ethylene glycol is less likely to bind to the enzyme. So ethylene glycol will not be metabolized to the toxic product and it will be excreted. Are you getting my point? So which statement describes why this treatment works? Ethanol is more likely to bind to the active side of the enzyme, and because ethanol is more likely to bind, it will prevent the binding of ethylene glycol, or we can say ethylene glycol is less likely to bind to the enzyme and is less likely to be metabolized to the toxic product. And because the ethylene glycol is not being metabolized, it will be excreted. The answer is D. Let's move on to MCQ number 24. Why do large increases in temperature or pH alter the enzyme activity? They change the three-dimensional shape of the enzyme. This is true. Large increase in temperature or pH will disrupt the tertiary structure of the enzyme, changing the 3D shape of the enzyme. They disrupt hydrogen and ionic bonds in the enzyme. This is true. The changes in pH break down hydrogen and ionic bonds. The changes in temperature break down the hydrogen bonds. They increase hydrophobic interactions in the enzyme. This is false because these changes, specifically temperature, they decrease the hydrophobic interactions and not increase them. The answer is A, 1 and 2. Let's move on to MCQ number 23. An unusual enzyme has been found in tropical grass. It catalyzes the hydrolysis of a fungal polysaccharide chitin into amino sugars. It also inhibits the activity of an enzyme in the locust guts, which catalyzes the digestion of amylose. What describes the action of this unusual enzyme? So guys, there's an unusual enzyme that is found in the tropical grass, and what it's doing, it catalyzes the hydrolysis of fungal polysaccharides chitin into amino sugars. So because it is catalyzing the hydrolysis of fungal polysaccharide, the polysaccharide contains the glycosidic bonds. So the reaction it catalyzes is the hydrolysis of glycosidic bonds because it's breaking down or hydrolyzing the fungal polysaccharides. It also inhibits the activity of an enzyme in locust guts, which catalyzes the digestion of amylose. So guys, if this enzyme is inhibiting the digestion of amylose, it means the reaction that this enzyme is inhibiting is the hydrolysis of glycosidic bonds, because the digestion of amylose also involves the hydrolysis of glycosidic bonds, because amylose also contains the glycosidic bonds. The answer is